So as it was said, so my name is Francis Rossignol. I'm a staff clinician at NHRI at the National Institute of Health. And I've been tasked to kind of summarize the developments that have been done for ManNAc principally, that we've been working on for more than 15 years now to develop at NIH. And I thought it was very interesting to look at some of the challenges that developing a therapy for rare disorder involve, and what are the steps that we've taken and some of our collaborators and other groups throughout the world have taken to address some of those challenges. And you've already heard many of them, so many of them will be repeats. It's an honor to follow so many people that do great science, and it's great to see that we are all aligned in our opinions. Must start with a few disclosures that given that NHRI has a patent on ManNAc and holds or has held several investigational new drug applications towards the FDA for ManNAc, and that we received some money from a pharmaceutical company named Leadion Biosciences to develop ManNAc at the moment. So the objectives here will be to summarize some of the challenges of developing a new therapeutic in a rare disorder, and to use the study of GNE Myopathy and the development of ManNAc as an example on how these challenges can be met. So to start right away with the challenges, we've heard just before Dr. Estevez talked about clinical trials and how they're important, and there are many steps that are important in developing a new therapy. Typically, a clinical trial, the goal is to establish both safety and effectiveness of a new drug using larger and larger populations and progressive phases. So phase 1 uses less patient, escalates doses. Phase 2 use more patient, goes more into safety, starts to do some more work, and then get to phase 3, a review for a formal application for approval, and then there can be extension studies in the larger population. All throughout the process, safety is a concern. So from the start, even preclinical, safety is a concern. So these is always something to keep in mind, but effectiveness of a treatment comes more and more as we progress through phases. So phase 1 is usually designed with very simple effectiveness outcomes, such as biochemical measurement. Phase 2 usually starts to integrate more of the biochemical measurements and some more clinical, although often underpowered to see the clinical side of it. And then phase 3 is really where our clinical outcomes and change to life come into play. But as you may already guess from this slide, there are many challenges to this, especially when you just look at the number of participants here in the typical chart of a clinical trial. So some of the questions that you can have is, what do we know about the condition? Do we have a therapeutic target? So that comes preclinically. It's, do we know what we're trying to treat? Do we know how it will work if we're trying to treat something? And what can we aim at? How do we measure effectiveness? So what do we know about the disease progression and natural history of the disorder? If we don't know how long it takes to have, for example, muscle weakness in GNE myopathy, how can we address if a treatment is effective in reducing the progression of muscle weakness or halting it? Is the disease uniform in progression or variable? So, or do we have to keep in mind that different individuals will have different rate of progression, which is the case in GNE myopathy? Do we know any clinical markers, biomarkers that are available to address the effectiveness? So, and are they sensitive enough to detect a change? So if this change is so small that it will take thousands of individuals to be detected, this is not a good outcome measure for a trial, at least not in a rare disorder. And that now brings a question of how many people will this require? So how do you find enough people? When we're talking about a disorder like GNE myopathy, it's usually involving multiple countries, travel. So that comes with the questions of funding at this point. And how do you manage to find your outcomes and make sure that you can detect the change with a minimal number of participants, because you will not have the thousands of people that are in typical drug that you see on the market. And then comes all the question of where will you find the funding for all this? Will you find the expertise for all this? And there are several elements that can help address these issues. So on the preclinical basis, bench work is very important. Basic science, mouse model, all that work is very important. For measure of effectiveness, natural history studies are very important, and they help in the development of new outcome measures often. And on how many people will this require? It often requires multicentric studies and alternative study designs using composite outcome measures that pull together many data together to make it stronger. And then as part of funding and expertise, the stakeholder involvement becomes very important to make sure that everyone's on board. So we'll start with some of the endeavor that we've taken at the NIH. I will preface that most of that work has been done by colleagues, so I've very lucky to have had a predecessor in my position as a principal investigator in the GNE Myopathy studies at NIH, Dr. Carrillo, and also colleagues on the basic science front, such as Dr. Husing, Dr. McLichten, and as well as Dr. Gulk, which is our section chief, that have a lot of experience with GNE myopathy, and most of that work come from them. So we'll start from the beginning, and I won't spend too much time on the basics of GNE myopathy, because I'm pretty sure you all know now what is the fact that's present in GNE myopathy. But it started, the association between distal myopathy phenotype and the GNE gene was first published in 2001 by a group in Israel. GNE is a bifunctional enzyme, so it accomplishes two functions in the cell. It transformed the derivative of glucose, UDP-glycanac, into ManNAc, and then has a kinase function that adds a phosphate group to ManNAc. And the ultimate goal of that pathway is to synthesize sialic acid that serves as a decoration on many surface proteins that, even though we call them decorations, they're very important for cellular functions. So it's not like it's a non-important step of the pathway. And from early on, the thought of supplementing the pathway either with sialic acid or some of its precursors. Okay. Why so? So there are many different hypotheses on how GNE myopathy actually affects the muscle. We are not fully sure of how the implications on pathophysiology yet, but some of it may be related to the lack of sialic acid and some of the proteins involved in the muscle, such as alpha-dystroglycan, which is an important protein that links the muscle to the extracellular matrix. And that has a lot of those sialic acid group. In fact, other disorders of glycosylation, such as Fukuyama muscle dystrophy, also involve the effects of the glycosylation of alpha-dystroglycan and can also cause myopathy. So here, in some studies that were done by an NIH group in the early 2000s, you can see that a normal patient usually has normal sialylation of alpha-dystroglycan, but in patients with GNE myopathy, this is very much absent, and we cannot see the cell walls as much as we can in the normal patients. Then there were development of mouse model, and I'm not a mouse model expert. I'm a physician. I rely a lot on my colleagues on the lab side to help me on that. But there are several models of GNE myopathy that have been developed and have shown some promises for ManNAc supplementation. One of them is the M712T mice, which was developed at NIH, where introduction of ManNAc in those mice that do have a phenotype that's a bit different from GNE myopathy. They have quite a severe kidney disease phenotype as well as a muscle phenotype. But introduction of ManNAc helps increase the survival of the mice. So if I take a bit of mouse, we can see that with low dose of ManNAc, there are no survival in the mice with the variant, whereas if ManNAc is supplemented a good amount, 1 gram per kilogram per day, there is significant survival. And now we know that there are ways to even increase further survival if, for example, the pregnant mice are given ManNAc. And that also restores some of the sialylation of the muscle. So here, the first square here, we can see a normal untreated mice, and next to it just a GNE Myopathy mice, where we cannot see any of the green SNA-lectin staining that's normally present when sialylation occurs. And with increasing dose of ManNAc, we can see that some of the sialylation of the muscle is restored, with some of the green staining coming back on the muscle cells. And similar results were obtained by Dr. Nishino's group in Japan with the D176B transgenic mice, where giving ManNAc increased significantly the survival of mice. We can see here in black, it's the abnormal mice that do not survive well. A low dose of ManNAc helps with survival, and for medium and high doses of ManNAc, it's so as they don't die throughout the study, it's at the same place as their littermate that are normal. And they also did interesting studies where they've shown that giving ManNAc to mice increases the distance that they can run, almost back to normal levels, and decreases muscle atrophy. Other work that has been done on the preclinical and involves development of new methods of biomarkers. And one of them is the development of quantitative methods of sialylation, so the presence of sialic acid on the surface of the membrane. And one of the techniques that was developed is the SNA-lectin quantification. So here on the left, we can see a composite image where we have a control substance, which is caveolin-3 in red, just to know where the cells are. And in green is the SNA-lectin that represents sialylation. And the more green there is, the more normal sialylation there is of the muscle. And here we can see other slides where a bit similar to in the mouse model, we see the similar phenomenon of absence of sialylation in the muscle, and it can be quantitated using computers to see the area and compare it to the control, which is caveolin-3 in red. So following this work is pretty much still ongoing on the preclinical front. So those are some examples of what has been done. But in parallel, we started studying clinically at the National Institute of Health Clinical Center the GNE myopathy itself. So bring patients in for different purposes. So we do have, we had 3 different studies. 2 are completed, 1 is still ongoing. So we have the natural history study that's still ongoing since 2011. We had the phase 1 and phase 2 ManNAc trials that went on from in 2012 and 2015. And we're expecting to start soon the pivotal trial of ManNAc as one of the centers involved in a multi-center trial with our principal investigator at Brigham and Women's Hospital, being Dr. Amato. As was said before, natural history studies are very critical in identifying outcome measures that can be used in a trial. So it is a very important step to make sure that we understand what is the disease progression to be able to assess after if we're able to affect that disease progression. So what we've been doing with our natural history study is a prospective natural history study. So we bring people in for 4 days inside the clinical center, which is a hospital entirely devoted to research. Initially, we brought people every 6 months for 2 years, and then annually. Now we're more going with a more relaxed schedule. And you can see on the sample schedule on the right, many of the evaluations we're doing. So we do questionnaires, we do patient-reported outcomes, history, physical examination, quantitative muscle assessment, and we'll talk a bit more about this particular measurement. Many other rehabilitation and functional evaluations, such as the AMAT, the 6-minute walk test, Jepsen-Hen function test, as well as cardiac and pulmonary function tests. So far, we've recruited 69 patients. 56 of them were admitted at the clinical center for one or multiple admissions to do all those studies. 13 patients were recruited only for questionnaires and questionnaire development. And one of the outcome measures that we've been using quite a lot for the development of trial is the QMA, so the quantitative muscle assessment, which is the machine that you can see on the left. So it's basically a fixed frame on which we put a strap, and the patient can force and pull against the strap, and the strength can be measured. And it has good normals defined, and we can see how much the muscle strength is preserved or affected. And using those measurements, having those sequential measurements over many, many years of the natural history allowed us to develop the disease progression model of GNE myopathy that you can see on the right. We know that GNE myopathy does not always start at the same age for everyone. It does not progress at the same rate for everyone. So the disease progression model defines a disease age where when knee flexion strength is at 50% of predicted, we define this as the 0. And we use that to be able to predict what is the trajectory of muscle strength and what is expected in 5 years, in 10 years, and so on and so forth. And that also allows us to associate with other outcome measures. So we've used many of the questionnaires that we deliver in the natural history. We see that the disease progression model and the disease age correlates well with some of the questions that we're asking. So the ability to walk, to climb stairs, fine motor tasks, handling utensils, dress self, the risk of falls, and many others. So having that intersection between a model to use in a future trial and many other outcomes is very important, because the more you can prove that your outcome measure that you're developing helps to predict other symptoms and development of other findings in the progression, the more solid evidence you have that your treatment is actually effective. So here we have at disease age 0, what would be the example where some of the muscle are in green, not very affected, and some more muscles are more affected. And then with disease progression, some of the muscle strength is lowered in the model, and it correlates with lower performance and in many of the other tasks. The disease progression model also includes a treatment parameter, so where we can introduce the treatment at some point and see if we're able to stabilize totally the progression, which we call the gamma of 0, or does it follow the natural history of the disease where gamma would be equal to 1. So the closer to 0 we are, the better the outcome of the treatment. And that will come back when we're talking about the different phases. All the work that we've done, and I won't spend too much time on it, but we've looked at other biomarkers and tried to correlate them to outcome issue. Here is one of the examples of it that was developed in the lab, the role of disialylated to sialylated Thomson fringed antigen, which is the ratio is decreased in patients with GNE myopathy, although this was not necessarily correlated to other clinical outcome. And then also look at the different GNE variants that can be described so that we have more solid evidence, and it becomes very important when you have a trial to make sure that all the variants you see are actually causative of GNE myopathy, that they are not variants of uncertain significance where you cannot know really if the person has a myopathy because of GNE or another gene, especially when you're going to give a treatment that target specifically GNE myopathy, you want to make sure that your population is as uniform as possible. The phase 1 of ManNAc at NIH occurred between 2012 and 2013, with the main outcomes being safety. So it was a single dose, placebo-controlled, with escalating doses of ManNAc to determine the better dosing and the tolerability of ManNAc. So patients were divided in 4 groups, either 3, 6, or 10 grams of ManNAc or placebo. It was well tolerated in all groups with no serious adverse events. Although in the group receiving 10 grams, there were some diarrhea and nausea, probably because ManNAc is a sugar and it's not totally absorbed in the gastrointestinal tract, and it attracts some water and feeds the bacteria there, so it probably causes some of the GI problems. Also, the higher doses caused elevated liver function test. But both ManNAc has been shown to be increased. So we can see here that after 2 to 2.5 hours, ManNAc peaks in the plasma, and sialic acid actually peaks as well. So the end product that we're really interested in, we can show that giving ManNAc is able to restore some sialic acid production in patients with GNE myopathy. ManNAc it's out of the plasma in about 12 hours. We usually say a half-life is around 2.5 hours, and it takes about 5 half-life to get rid of a small molecule. And sialic acid peaks after about 8 to 11 hours and is still present 48 hours after ManNAc is given. Using all those data, we were able to come up with a suggested dose for future trial, which was 6 grams twice daily, as 10 grams had more side effects and did not seem to raise more the sialic acid levels compared to 6 grams. When you look at the data in blue, you can see that actually the peak is higher for 6 grams a day than 10 grams a day. So for all these reasons, 6 grams twice daily seemed a better dose. So came the phase 2 trial of ManNAc between 2015 and 2018, where 12 patients were enrolled, some for 3 grams twice a day, and then when tolerated, escalated, and because of good tolerance, 6 grams BID was initiated with 6 patients. And the primary endpoints here were mainly biochemical, so prove that biochemically ManNAc works. So it was mainly muscle biopsies, sialic acid concentration. And then clinical endpoints were secondary. So it's not enough patients to be able to see if there are clinical outcomes changes, but trying to address for the first time, do we have a tendency that it shows that it's promising for some of the clinical outcomes? So the first endpoint was at day 90, but there were study extensions up to 30 months where a lower number of patients participated, with 8 patients going through all in all study. And we can show with that data that after 90 days, sialic acid production is restored at least partially in the muscle. So here we have a muscle at baseline and the same patient 90 days later on ManNAc. We can see that there's quite a lot of red and not a lot of green on the left-hand side, but on the right-hand side, there's quite a lot more green. Say, well, what if it's only randomness? Is it really more? Is it just my eyes tricking me? And that's where the importance of the preclinical work, where developing quantitative methods came on handy, where we can prove using those quantitative methods that it's significantly higher in patients that did receive ManNAc. As I was saying before, the study was not powered enough to see if clinical outcomes were better. There was not enough patients to be able to prove that, but we still looked at the data to see, do we have some hints that it may be effective clinically? And we do actually have some hints. So when you look here, it seems like a busy slide, but on the upper left-hand side on A and B, see that the lower extremity LE strength and the upper extremity strength has pretty much stabilized in patients that receive ManNAc over 18 months. You can see it do not have points for 24 and 30 months for the upper extremity strength because there was a problem with the machine, so the data was unreliable. You can see that other outcomes such as the 6-minute walk test, the human activity profile in DNE, or the IBM-FRS, which is another patient-reported outcome, did not show significant difference between the natural history and the patients interested in ManNAc, but at the same time was not powered to do so. So we have some encouraging signs there. And I'm bringing back the treatment parameters that I introduced a bit earlier with the disease progression model, where you can see that in the period where we have good data, so at 24 and 30 months, because we have less patients and because the upper strength cannot be used, this data is probably more reliable. But in G and H, at 12 months and 18 months, you can see that the treatment parameter has a gamma of around 0.5 to 0.6. So a curve that would be around the third, if I start with the one on the bottom. So a slower disease progression, and actually at 18 months reaches significance. So it's very promising for the future studies, but as I was saying, underpowered because not enough patients. So where are we now? We are now planning to participate in a trial, a pivotal trial, with the goal of bringing the drug to FDA for approval, which is called a MedGen trial. It's coordinated by the NeuroNext network that's financed by both NINDS and NIMS, which are two other NIH Institute, NHRI is part of Social for the clinical center, and as well as 9 other centers in the US that you can see, and our industry partner, Leviant. 51 participants will be recruited across the US, 18 patients at the NIH Clinical Center, including international patients. And the study is going to be 2 years plus extension. So people are going to be seen. We need to make sure that they are eligible for the study. So between 18 and 70 years old, have a molecular diagnosis, so a genetic confirmation of GNE myopathy, which we can help obtain if people do not be able to communicate in English because of all the questionnaires that we're giving, able to travel. And then there are some outcome measures. So we want to make sure that the strength QMA is within a certain range, that we can make sure that we are sensitive enough to detect a change, because if the strength is too low, we will not be able to detect a change. That people did not receive ManNAc or sialic acid before, because that would be confounder, and to make sure that the labs are all within safety limits, so that we are not having another problem added to ManNAc to confound if ManNAc is safe or not. And people will come every 6 months for visits. Up to 2 years are actually at the end of the 2 years of the last patient that's recruited. So some patients may actually go to 36 months study with calls every month to make sure there's no side effects and so on and so forth. If you want more information, I provided the link for the full clinical trial there. One of the questions will be, will I receive ManNAc with this? You'll see that the design is that we need to have some patients that do not receive ManNAc to be able to have a comparison. So the randomization is going to be 2 to 1. So 2 patients out of 3 will receive ManNAc and 1 out of 3 will receive placebo, in order to be able to better address if ManNAc is working or not compared to receiving nothing. This is pretty much all I had for today. I thank you very much for all your attention. I thank many of the people that help us with this, all the patients and families that came to the clinical center that help us with all those studies, all the NIH team, my predecessor as the PI, Dr. Puriel, that did tremendous work before, Dr. Gall, Dr. Fusing, Dr. Malik, Dr. Leoclac, that did tremendous work before, and all our collaborators outside of NIH. And I provided the email if you're interested in our work, either the natural history study or the future trial. You can communicate, and one of us in the clinical team, either me, Andrea Bowling, Jessica Jang, or Akili Nguyen, can answer some of your questions. And I'll be happy to answer any questions you have.